Here we go. We start again. Oh my goodness. So thanks to Dr. Kim and the organizing committee for inviting me to introduce you the work that we are doing here in Moncton, New Brunswick. So we are located on the East Coast, so eastern than Quebec province, and just before Halifax. So for those of you that know Nova Scotia. So I'm located, as mentioned, at the University of Moncton, a French university outside of Quebec. And my work over the years has been focused mostly on VHL and kidney cancer. And today I chose to present a project that has been funded by the Canadian VHL Alliance in 2020 during the pandemic, and that we are very excited to start. It's a beginning of the work, but we are glad that we were able to make it with the funding. So I don't have any disclosure to mention. And of course, the good thing about this meeting is that we don't have to introduce the VHL disease and the importance of studying this disease, this rare disease, and on a basic and on a clinical side. But it's also difficult to present, to introduce this subject, because this audience is expert in the fields over the years and many years. But anyway, I chose, I don't think you will learn a lot of things in my next 5 slides, but I chose to present them because it's the way we develop the project. So as mentioned earlier and very well explained by Dr. Leinard, so the VHL disease is a familiar and rare disease that affecting similarly men and women of all ethnicity. It's characterized by germline mutation occurring on the VHL genes. You can have deletion or missense mutation, and this mutation leads to different clinical manifestation in patients, and which lead to classification of the disease mostly in 2 types, so type 1 or type 2 disease based on the absence or the presence of pheochromocytoma. Furthermore, the type 2 disease has been further subdivided and classified by the high risk or low risk of developing RCC. Again, we know that the VHL gene is located on the chromosome 3p in a region where other important tumor suppressor genes are located and involved in the development of kidney cancer. The gene is coding for a protein of 213 amino acid, and we know that we also have 2 other shorter isoforms. But all of them have tumor suppressor function. To date, we know that more than 1,500 mutations have been identified on the VHL genes. And again, as it has been explained, and I think all of the audience is very familiar and expert in this field, so we know that the most characterized function for the protein VHL is E3 ubiquitin ligase activity that gave him the opportunity to bind the hypoxia-inducible factor, so the F1 or the F2 alpha, not present in the slide, but we know that from their discovery. So we know that the prolyl hydroxylase and the VHL association with the F alpha subunit give in the targeting for the proteasome destruction, but during hypoxia or during VHL inactivation, we are in presence of stabilization of F alpha, which leads to development of tumor and angiogenesis through VEGF and PDGF as well. Moreover, several other groups identify other function for VHL that has been not related to F, and our lab and others are focusing on the role of VHL in lysosome and endocytosis and autophagy through microRNA shown in 2012, and my team is more focused on autophagy and lysosomal dynamics. So all of this basic research over the years lead to the development of a new therapeutic approach. And you can see that on this slide that is ending in 2019. And since then, we have all of development with the F2 inhibitors that is not on the slide, but that we will hear during today and tomorrow, for sure, during this conference. I started to work on VHL during my PhD and mostly during my postdoc at Stanford with Dr. Jecha. And at that point, we are investigating if we are able to find small molecules to sensitize the cells that are mutated or inactivated for the VHL genes, mostly in clear cell renal cell carcinoma, of course. So this work led to a paper and identification of the STF62247, as mentioned by Dr. O in the introduction. So with this small molecule, we are able to show that we are able to target the mutated VHL cell compared to the cell that have the functional gene. So this cell, you can see that over the increased concentration, we have a decrease in cell survival, and that was true in vitro and also in vivo in the 786 xenograph model, and also in another SN12 model with a shRNA for VHL. Since then, we developed other models. So we we did the rescue experiments where in the positive VHL cell, we use the CRISPR-Cas9 to remove the VHL that has been introduced, as you can see on the Western blot on your left. So we did that in the RCC4 cell line, RCC10 and 786 cell line. And when we remove the expression of the VHL, we were able to sensitize the cell to the STF62247 in a similar way than the parental cell line. We also went to look for the specificity of the molecule. We have a look to other cancer cell type. So we went for colon carcinoma, as well as pancreatic and prostate cancer and lung cancer, where we see the presence of the VHL protein inside the cell line. And we did a cell viability assay. And again, as we increase the concentration of the small molecule, the STF62247, we don't see major cytotoxic effect in this cell line compared to our kidney cancer cell line with the loss of VHL. We also did the xenograph model with the HCT116, and we don't see any significant effect on tumor growth. When I started my lab, we are investigating which pathway, which molecular and proteic pathway that are involved in toxicity of the STF62247, and we performed a SILAC proteomic assay with Nadia Bouhamdani, who started a master, a PhD, and a postdoc in my lab. And we identified several proteins that were deregulated in the signalization of the small molecule, but most of them were related to autophagy and endocytosis pathway. So we look for the implication of this pathway, and using CRISPR, we remove Becklin or ITG5 in our cell line, both involved in the autophagosome process and the beginning of the autophagy pathway. And as you can see, we were able to rescue most of the viability of the cell treated with the STF62247. A phenotype that is very interesting when we treat the cell is the presence of this large intracytoplasmic vacuole inside the cell with the loss of VHL. And we knew from the beginning that it was not linked to apoptosis. We show it in a paper, and then that lead us to autophagy, endolysosome, and lysosomal pathway. So in another paper, Nadia developed a lot of molecular tools to study the autophagic pathway. This pathway is very dynamic. So in sense of autophagosome is sequestered organelle and misfolded protein and long-lived proteins for targeting them for degradation through the lysosome. So they need fusion with the lysosome for proper degradation. So it looks like a garbage process. So we need acidity from the lysosome or endolysosome to degrade the organelle or the protein. So by developing and using tools that were in the literature, we were able to show that our small molecule was not necessarily inducing the autophagic flux, but instead it was blocking the fusion between the autophagosome and the lysosome. So this is why we are seeing a high number of autophagosome present in the cell and this large vacuole. We quantify all of this, and I will try to resume here in this slide where over time of treatment, we see an increased number in autophagosome in the cell with a loss of VHL compared with the cell that have the functional genes. And if you look at the lysosome, it's a reverse thing. So we don't have enough lysosome to degrade all of this autophagosome present in the cell. And we have fusion, but the cells are not able to recover and degrade the machinery and probably will die from it. If we compare with the VHL cell, we have a high number of lysosomes. And as you can see, the number of autophagosomes significantly decrease over time. So we also continue to characterize this process by using a different dye as a lucifer yellow that is staining the endolysosomal organelle compared to the lysotracker red that is more specific or sensitive for acidic organelle. And what we saw and when we show in this paper is that the STF62247 cause a swelling of the endolysosomal process in green here. So, and this correlates with a lot of large intracytoplasmic vacuole that we see inside the cell. So we have fusion in it, we have movie to show it, and then the cell just die and not able to recover. That brings us for a number of years to identify what is the target of the small molecule intracellularly, what is happening there. And we use different approach to do it. And one of them is we develop a resistant cell line for the STF62247 and use the next generation sequencing. And the other approach that we use is that we take advantage of the autofluorescence of the STF62247. So we migrate protein on a nature gel, and we incubate the gel with the STF drug solution, and we cut the band where the drug is binding to the protein, and we send the band for MS analysis. From all of this analysis, we found VAC14 and PIkfyve as a potential target for the STF62247. And because PIkfyve is a kinase, we also did the kinase assay. We send for a kinase assay outside, and they show us that the STF is binding the lipid kinase PIkfyve at nanomolar concentration. So we're kind of very excited about this work. At the same time, we know that there are also other PIkfyve inhibitors that are coming in the cancer field. So we try to investigate that in the way of the kidney cancer cell. So as mentioned, PIkfyve is a lipid kinase, and what is doing inside the cell is the only enzyme that is able to phosphorylate the phosphoinositide 3-phosphate into PI3-5P2, which is a very low abundance phosphoinositide present at the lysosomal membrane. But although it's very low abundance, it regulates the lysosome dynamics and size, fission and fusions event, as well as lysosomal reformation. And paper show that PIkfyve inhibition impairs autophagic flux and cause enlargement of lysosome, leading to non-apoptotic cell death. So that makes sense with what we have in our hands with the STF. And then we further pursue this investigation using other PIkfyve inhibitor, as I mentioned, the YM that is really known, the Apilima that is also not discovered for its PIkfyve inhibitor, but now is investigating in, I think, lymphoma for its benefit effect, as well as vacuolin and APY. So using, again, the RCC4 cell line, but we also have the data with other cell line and we show that the vacuole present inside the cell are similar between STF and all of this PIkfyve inhibitor and are bigger in the cell that have a loss of VHL or mutation on VHL compared to the VHL wild type. And these cells are also sensitive to all of this inhibitor, at least the cell with a loss of VHL. We use an antibody to measure the phosphoinositide level inside the cell. And as we use immunofluorescence because it's very difficult to quantify. At this point, what we see, it's a decrease in the phosphoinositide PIkfyve P2 as expected when we treat with the STF, observed mainly in the RCC4 VHL mutated cell. So with this project that I wanted to talk about, and I think I have to move fast, we want to know whether it could be a possibility that VHL mutation associated with VHL disease can be targeted through the PIkfyve inhibition using STF or using other PIkfyve inhibitor. So we propose in this project to generate an in vitro model of VHL mutated cell associated with VHL disease, test the potential of PIkfyve inhibitor to sensitize this model, and to study autophagy and lysosome dynamic. So we have decided using mutagenase, this kit that we probably all know, to create missense mutation into the PIkfyve vector, VHL PIkfyve plural vector. So we chose about 10 mutation points that are known in the literature to be hotspot mutation or present in more patients. So we generate this point mutation, and we introduce them in the 796 model. We chose the 796O cell because they don't have VHL, they have a stop coding, so they don't express the protein. So this is why we chose the 796 cell, and we do clone selection. We just located all of the point mutation we have decided to work with, and we classified them between type 1, type 2A, B, and C disease. Here we have, after selection and clonal selection as well, we pick one clone and do a Western blot, and we also do sequencing to make sure we have the right thing in our cell. We have difference in the VHL expression, of course, because it's related to the VHL activity, and this is corroborate with the IF2 alpha level inside the cell, which is associated with the disease as well. Then a master student in the lab, they took all of this cell line and treat them with at least 5 PIkfyve inhibitor. We present the data by type of the VHL disease, type 1, type 2A, type 2B, and C. So if we compare with the empty vectors, so the cells that don't express VHL, then by using STF62247, again, we see a decrease in the survival of the 796O cell, which is comparable to the point mutation we choose for type 1 disease, and also for type 2B disease, which is associated with development of clear cell renal cell carcinoma. Otherwise, we also choose 2-point mutation for the type 2A disease. And as you can see, the cells are not sensitive. It's similar to the wild type that is not presented here, but we have the data in the lab. But we have significant difference where the cells are very less sensitive to the STF62247. And it's also observed with the point mutation, the leucine 1882 valine, associated with type 2C disease that are not sensitive to the STF and also to other PIkfyve inhibitor. Here we chose to present the APY201, which is an analog of apilimumab. These cells are more sensitive to this inhibitor. First of all, the concentration is very low, but we have significant difference for sensitivity with this PIkfyve inhibitor. It's not perfect. We still need to investigate and understand all of this data, but the data suggests that some point mutation associated with the development of clear cell renal cell carcinoma could be more prominent for the PIkfyve inhibition. We also look at the VHL stability in response to at least the STF62247 using the cyclohexamide protocol, as some of you know, and has been established and published already. So as we can see, yes, we have a decrease of stability of the protein at 63% in the control right type. And if we compare for that, we see that the point mutation associated with type 1 disease, with the stability of the protein is affected by the mutation point of the VHL. This is also known from the literature. And some of them are more stable than others, sorry. And when we treat with STF, we have a significant difference on a lot of them associated with the point mutation. Finally, since I'm almost done, we look at the lysosome. We look at the lysosome number and distribution inside the cell. We use confocal microscopy, we use Western blot, we use activity analysis. And first of all, what we see is that between the point mutation associated with the VHL disease without treatment, just the point mutation, interestingly, we see a very nice difference on distribution and number of lysosome between cells that don't express VHL and the cells that express the VHL wild type. And then if we go through the different mutation type, here it's 2C and 2A, and here it's type 1, we also are able to see some difference more associated with the wild type or the empty vector, depending of the point mutation. More recently, we treated the cell and did the same exercise with the confocal microscopy to look at the lysosome. And again, we see a nice difference between the control, but also very elegant difference when we treat with the drug, where the lysosome seems have a low abundance when we treat with the drug in the cells that don't express VHL or if they express the type 1 disease compared to the 2A or the 2C. So altogether, we were able to generate cell models that are associated with 10 germline mutation found in patients with VHL disease. And our results suggest that the STF or PIkfyve inhibitor can sensitize this model differently, which could be related to protein stability or to other processes like lysosome dynamic. On the next step, we will investigate the autophagic flux and other functional studies and on PIkfyve activity as well. And we can have a look to combinatory treatment with agents that are currently used in clinic for VHL disease or kidney cancer. And we would also interested to investigate the PIkfyve expression in patient tissue with collaboration, of course. So this is ending my presentation. I would like to thank, of course, the funding, the Canadian VHL Alliance and the Cancer Research Society for this project, as well as all lab members Nadia, Dominique, and Chloe that are working on this project, as well as other collaborators that we have developed over the years. So I will be happy to take a few questions.
Thank you, Sandra, for that wonderful talk. Maybe we could just stop sharing the screen. Great. Any questions from the audience? Why don't I just start something off, Sandra? Your talk actually reminded me of an old study that sort of linked VHL HIF function to the endocytic cycle. So under hypoxia, things tend to slow down. And lysosomal data sort of remind me of that. That's number one. And I was wondering if you could comment on that. And the other is the link, of course, HIF with BNIP3, BNIP3L, which are genes involved in autophagy. So maybe the first question, the potential link to the endocytic cycle itself.
Yes, I think it's your team that published a paper on idocytosis role with IF, with Rabaptin 5 and Rab5. We didn't pursue this work. When we look at endocytosis pathway in our hands with our compound, it looks like independent of IF alpha 1 or 2. We use shRNA at the beginning, and after that we try with CRISPR. We remove the 1 and we remove the 2 alpha, but we were not able to remove both. So, there is still some potential that one compensates for the other. But in our hands and in hypoxic condition, we don't see major difference on lysosome or endocytosis process.
Okay, great. Thank you. I see Dr. Kim has his hand raised. Billy, I see you're feeling better. Good to see you.
Yeah, very nice talk, Sandra. So I had a quick question. Do you think that the effects you're seeing are somehow specific to renal cell carcinoma, given the links to autophagy? I remember Bill Kalin had published a nice paper about 5 years back about P62 being amplified on the 5Q arm. So I was wondering if you thought that there was any specific lethality to kidney cancer rather than other tumor types.
We think about it, and also we developed the model in the kidney cancer cell lines. So with the VHL point mutation. So of course, that could be all related because we know that this tumor can be dependent of the autophagic flux. We were questioning ourselves if other tumor type that are dependent of autophagic could be used in our approach, at least to see if we have difference or if we have an effect. But we haven't looked more carefully into it.
Okay,